All right, in this video, we're going to do an introduction to the anatomy and physiology of the ear. That's going to include the hearing, auditory side of what the ear is doing, and also the vestibular system, which is the equilibrium or balance side of things. So first, some quick anatomical distinctions. The ear is split into separate external, middle, and inner ear cavities or areas. And the easiest way to show how this is split apart is by starting with this tympanic membrane. The tympanic membrane is what you probably refer to as the eardrum. And it forms a barrier that is probably the easiest way to think of. Outside of that, we'll call that external ear. Once you get to the tympanic membrane, everything from there inside of this bony cavity, we'll call middle ear. And then once you're completely inside the bone, this is all inner ear. So all of our actual mechanoreceptors, right? The cochlea is this sort of snail-shaped organ responsible for hearing. And then these semicircular canals and a few other structures, that's the vestibular side of things, right? And then the nerves coming out of the inner ear, the vestibular and cochlear nerves, they merge to form cranial nerve 8, which is the vestibulocochlear nerve. So it's carrying both sets of information. Hearing from the cochlea, and then balance and equilibrium stuff from the vestibular nerve. Right? In that middle ear cavity, you also have 3 very, very small bones. These are called ear ossicles. And those bones, the malleus, the incus, and the stapes, are really just transmitters. So when sound waves come in and the tympanic membrane vibrates, it's going to transmit those vibrations into the bones, and then the bones will transmit them into the inner ear. So you can basically amplify your sound waves onto the cochlea and get better hearing sensation. All right, and this diagram is trying to show that in a bit more detail. So first off, remember that sound waves are just vibrations. When an object vibrates, you get pulses of high and low air pressure. So for example, someone hits this tuning fork. Whenever the tuning fork vibrates this way, it's going to compress the air molecules, and you're going to get a region of high pressure. And then when the tuning fork vibrates back this way, it's going to create a vacuum, and you're going to get an area of low pressure. And so the hearing mechanism itself is really based on mechanoreception, right? When these pulses, these vibrations of high and low air pressure make their way into the ear, you're activating mechanoreceptors that are being vibrated. Those are what you're going to find in the cochlea. And so all of this is just the basic physics of sound, right? And there are a couple different properties of sound that you can differentiate. Those properties are frequency and amplitude, right? Frequency is asking how many vibrations are there per second, right? So this set of units, hertz, really just means vibrations or cycles per second. And really, really low pitch sounds are, down into the double digits of hertz. And children can hear as high as 20,000 hertz. That number tends to diminish pretty quickly with age. But what you hear as pitch, like a note on a guitar, or someone with a high voice versus a low voice, that is really just your brain's interpretation of frequency. Right? And so pitch for sound is really a lot like color is for vision, right? Amplitude, on the other hand, is how strong or powerful the sound wave is. And on a graph, that would be like the height, right? This would be like a low amplitude sound. This would be a high amplitude sound. And so stronger, more powerful vibrations mean more amplitude. That is perceived by the brain as how loud the sound is. And we measure that in decibels. And so just remember, you can have any combination of pitch and amplitude. You can have a high-pitched sound that's quiet, or a low-pitched sound that's loud, or the other combination, because they're 2 completely separate properties, just like brightness and light for the eye. You can have varying brightness and variations in light that are completely independent of each other. Okay, so let's talk a bit more about the anatomy of how this actually works. Right, so this cochlea, right, it looks like a tube that's rolled up. It ends up looking like a snail shell. The reason it's rolled up is just to save space. If you unrolled it, it would be poking into the middle of your brain. And if you cross-section just one little section of that tube, you get something that looks like this, that actually looks like 3 tubes within a tube, right? So these scala vestibuli and scala tympani are just surrounding fluid that are sort of propping the tube open. All the mechanoreception happens inside of this cochlear duct, that's the middle one. And very specifically, this organ of Corti, this is where the actual sensory receptors are, right? So that organ of Corti, that's what we're blowing up, like a zoomed in illustration down here. And what you find inside of it are these hair cells. And hair cells are just mechanoreceptors, right, that have these structures called stereocilia. They're not really true cilia, but they kind of look like it. And those stereocilia, those are your mechanoreceptors. They're attached to this very, very thin sheet of connective tissue called a tectorial membrane. And when sound waves come in, the tectorial membrane is going to vibrate up and down. Think of imagine like a sheet on a clothesline on a windy day, just whipping up and down. So the harder and more like higher amplitude the sound waves are, the more vibration you're going to get in that tectorial membrane, right? That is going to pull on those stereocilia, and they have mechanically gated ion channels, right? Ion channels that open when there's mechanical force, right? That's the vibration. And so if you have a sound come through and vibrate the tectorial membrane, we open ion channels and will depolarize these hair cells, right? At the bottom of the hair cell, these little yellow things are showing you that they're connected to the cochlear nerve. And so the louder the sound is, the bigger the vibrations are, the more ion channels you're going to open. You expect a higher frequency of action potentials going back to the brain, and that is how your brain knows that a louder sound is taking place. All right. Okay, so that explains the amplitude side of things, right? How loud a sound is. The next issue is detecting pitch. How do you tell the difference between low notes and high notes? And so in this illustration, what we're doing is they're imagining what the cochlea would look like if you unrolled it. So in reality, right, your cochlea is the snail shell organ up here. We're just saying, all right, pretend that we unrolled it into a straight tube. The way it's set up is the beginning of the cochlea, the base, has very, very tight, short connective tissue fibers, and they resonate really well with high frequency sound, right? So resonating frequency really just means the natural frequency that an object vibrates at. So when you pluck a guitar string, the note you're hearing, that is the resonating frequency of that string. And of course, the shorter, thinner, tighter ones make high notes, and the longer, thicker, looser ones make low notes. And so your cochlea is basically like a reverse string instrument. You put the little tight strings over here, and when the sound wave comes in, they resonate, they vibrate better because that's their natural frequency that they vibrate at. As you move down the cochlea, the connective tissue fibers, right, and the basilar membrane is just the membrane that the hair cells sit on top of. The connective tissue fibers get longer and thicker, right? And so just like a longer, thicker instrument string, they respond best to low frequency sound. All right, and so your brain basically has a map and says, if the action potentials are coming out of the base of the cochlea, must be a high frequency sound. If it's coming out of the far end of the cochlea, must be a low pitch, low frequency sound, right? And that is how your brain determines pitch, right? And of course, everything in the middle is possible too, because you're pretty good at knowing, even if someone's just like a very like half a note off in a song, you'll know that it sounds out of pitch. And so your brain is very, very good at using this system to determine the frequency of sound waves. All right, that was the basics of what's going on in the ear. The last little bit of this is going to be on the vestibular system. And the vestibular organs, there are a handful of them. These two, the macula, which the utricle and the sacula are both examples of macula. They're these structures that are actually incredibly odd. You have hair cells, right? So it's still mechanoreception, but on top of the hair cells, you have this otolith membrane. And otoliths is really just a very fancy word for little rocks. They're actually tiny little stones made of calcium carbonate, and the whole purpose is to give some weight, right? So when you tilt your head forward or back or side to side, gravity pulls those otolith stones in whatever direction gravity is carrying them. That activates these hair cells, and you get signals back to the vestibular nerve telling your brain that your head's moving. And so this is body positioning. This is how your brain knows if you're tilting too far to the side, starting to lose your balance. And it's these two, the utricle and saccule, are specifically detecting linear acceleration, right? And we say acceleration because it's not just movement, right? If you're sitting in a car on the highway and your cruise control's on at 60 miles an hour, you're not changing your speed, and so these won't be activated. You would only turn these on if you hit the gas or hit the brakes or do a turn, and that'll make them shift in that particular direction. All right. The other set of vestibular organs are semicircular canals. The crista ampullaris is the actual sensory structure, right? That's basically this entire thing. And the semicircular canal, they didn't draw it completely in this figure, but the semicircular canal would loop around like this, and the canal itself is just filled with fluid. So what ends up happening is if your head starts spinning, like think spinning in a chair and you're getting dizzy, all of these hair cells shift, right, the opposite direction that you're spinning. Just like if you, grab a flag and run into the wind, the flag will go behind you. If you spin a particular direction, like in this example, the person would actually be spinning their head that way to make the hair cells move this way. Again, that activates axons going to the vestibular nerve. Your brain knows which way you're spinning, and the feeling of being dizzy is really like when the chair stops spinning, the fluid still moves a little bit in the canal for a few seconds, which is why you get the perception that you're spinning, but you actually aren't, right? And so your feet try to compensate and you fall over because you're not getting a correct set of information about the world around you. All right, so that's the end of my ear review. As always, hope it was helpful, and this will be the last video in this section going over peripheral nervous system.